It is just past sundown on the eastern shore of San Juan Island in the late summer of 1961. The water in the bay has gone the color of slate. The pebbles along the Wrack line are wet. The kelp is breathing softly with the tide, and in the shallow water at the foot of the dock, there is a small, near-transparent shape pulsing in the half-light, no larger than the palm of your hand, almost impossible to see at all unless you stand very still and let your eyes adjust to the gathering dark. It looks empty. It looks like a piece of moving glass. It looks like nothing. Then, very faintly, a thin ring of pale green appears around the rim of its bell. It pulses once. It pulses twice. The color is so soft you can almost convince yourself you imagined it. And then it is gone. And the bell is just a piece of moving glass again, and the tide carries it gently back out into the cold. Welcome to Midnight Volumes. Tonight, we sit with the crystal jelly of Friday Harbour. You are about to spend a long, slow evening with one small animal and one small protein. Both are made almost entirely of water. Both spend their lives at the level surface of the sea. Both, for most of human history, would have been beneath notice. And both, very quietly, have changed the way biology sees itself. The jellyfish has a name. It is Aequorea victoria, the crystal jelly, a hydrozoan medusa that drifts along the cold Pacific shelf of North America, from the Bering Sea to Southern California, particularly common in the deep, cold inlets of Puget Sound. It was first described in 1902 by two researchers named Murbach and Shearer, and given a Latin name that itself carries the smell of seawater. Aequorea, from equor, the Latin word for the level surface of the sea, the calm plate of water on which the medusae rise and rest. It is, in other words, named for the place where you would find it, and for the way you would find it. Floating, suspended, almost exactly at the line between deep and shore. The place is named too, Friday Harbour, on the eastern side of San Juan Island, in the upper San Juan archipelago of the state of Washington. There has been a marine field station here since 1904, run by the University of Washington and called, simply, Friday Harbour Laboratories. It is one of those places where biology happens by accident, because the water is right and the boats are small. People come in summer to study things that live too softly to be studied anywhere else. Soft-bodied invertebrates, plankton, tube worms, and jellyfish, of course, in great pulsing constellations, especially in late summer when the medusae are old enough to glow. We are going to spend most of tonight with one man, 3 molecules, and a little less than 50 years of patient work. We will not raise our voices. We will not rush. The story we are about to tell is gentle and a little strange, and it earns its silence. This is one of those quiet stories where almost nothing dramatic happens out loud. There is no shipwreck, there is no extinction event, there is no asteroid. There is only a series of small, careful observations made on cold mornings by people in rubber boots on a dock that smells of low tide. There is a man with a notebook. There is a sink. There is a bucket of seawater. There is a long, slow patience. And at the very end of all that patience, there is a kind of light that biology has been trying to put its finger on gently since the time of the Greeks. Not a chemical glow, not a phosphorescence, a specific, repeatable, traceable, modifiable green light encoded in a single gene, exportable to almost any living cell that can build a protein. A lantern, in the end, that biology learned to hold. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too. The story begins almost in a kitchen, or a sink, or a back room of a small wooden building at the edge of the bay. It begins with a man named Osamu Shimomura, a Japanese organic chemist who, as a 16-year-old boy in Isahaya, 25 kilometers from Nagasaki, was briefly blinded by the flash of an atomic bomb, and who 20 years later began crushing jellyfish in galvanized buckets on this very dock in order to understand why they glowed. It begins with a small protein that in the dark can convert blue light into green. It begins with a gene that, at first, almost nobody could find a use for, and that today is in nearly every molecular biology laboratory on the planet, written into mice, into worms, into fish, into bacteria, into corn. It is a story that finishes in Stockholm on the 8th of October 2008 with 3 men in suits and a Nobel Prize in chemistry, and one fourth man who, by then, was driving a courtesy shuttle bus in Alabama. This is the lullaby version of that story, not the textbook version, not the documentary with loud music version, just the slow, grateful version. The one for ears that are tired, the one for a mind that wants to hold a small green light in the dark and let everything else go. Let us start where the molecule starts, not in a laboratory, not in a paper, in the water. The crystal jelly, when it is alive, is almost entirely transparent. Larger specimens grow to about the size of a saucer. The bell is a thin, gelatinous dome, ringed by uneven tentacles that may number as many as 150 in a fully grown adult. Up to 100 fine radial canals run from the center of the bell out to the rim. The mouth is contractile and lives at the center of the bell on the underside, a manubrium that can stretch open wide enough to swallow another animal half its own size. The whole structure swims by gentle muscular contraction of the bell, a slow inhalation, a slow exhalation, a soft pulse against the cold of the Pacific shelf. In life, you can almost see straight through it. The jelly bends light only the smallest amount. You can read the shape of a kelp blade through its bell. You can see the shadows of the boat above through its body. It is, in the literal sense, a creature shaped to be invisible. And yet, this transparent thing, this water inside water, has, on the underside of the rim of its bell, in small concentrated granules, 2 molecules that between them can produce light. The first is a photoprotein called Aequorea. It is named, like the genus, after the level surface of the sea. When a calcium ion drifts into the right pocket of this protein, the protein undergoes a conformational change, and a small molecule trapped inside it releases its energy as light. Blue light, a wavelength of about 465 nanometers, somewhere between deep ocean and a cold winter sky. The second is a small protein called the green fluorescent protein, abbreviated GFP. GFP does not by itself make any light. It is not a photoprotein. It is not a luciferase. It does nothing if you put it in the dark and leave it there. It is, in its way, a kind of biological optical filter. It absorbs blue and ultraviolet light, and when it does, it lets that light back out as green. In the granules at the rim of an Aequorea bell, these two proteins live next to one another. When calcium triggers equorin, the equorin makes blue, and the GFP passes it back out, very softly, as green. That is what you are seeing on a cold dock at Friday Harbour in late summer, when a small, near-transparent thing in the Wrack line pulses pale green twice and goes still. That is a calcium signal becoming a blue photon, becoming a green photon, in a molecule that nobody on the planet in the late summer of 1961 has ever taken apart and looked at directly. The world has the green light. It does not yet have the explanation. We are sitting at the very edge of that explanation tonight. We are in 1961. The bucket of jellyfish is on the dock. The man who will spend the next several decades of his life understanding what is in those buckets is upstairs, in a small room at the field station, sharpening pencils. There are about 1 million jellyfish in his future, and, although he does not know it yet, a Nobel medal as well. But none of that is the point of tonight. The point of tonight is the thing in the Wrack line. The point of tonight is the soft green light twice, then nothing. The point of tonight is the strange, gentle fact that biology of all things, with its 10,000 textbooks and 10 million experiments, biology only learned how to glow because somebody noticed a creature that was glowing already and was patient enough to ask why. The wind across the bay is light. The tide is going out. The light is going. Somewhere over the eastern hills, the first stars are beginning to come out, very faintly, behind a thin layer of late summer cloud. The lab boats are tied up. The buckets are full. The jellyfish drift in slow, patient circles in their seawater, doing what jellyfish do, which is mostly nothing, gently, with grace. We are going to spend the rest of this evening following that small green pulse. We are going to walk it from a bucket on a dock to a structure inside a beta barrel, to a worm in a petri dish, to a Stockholm lecture hall, to a microscope room in a hospital where surgeons are trying to make cancer cells glow. We will move slowly. We will name names. We will be quiet about it. For now, take a breath. The dock is cold. The kelp is wet. The tide is going out very slowly, the way tides do at the end of August in the northern Pacific. There is a bucket of seawater at the end of the dock with a few medusae in it, drifting in slow circles. They are ordinary. They are unremarkable. They are gelatinous. They are small. They are about to break biology open. You will hear about them again. By the end of tonight, you will know the names of the people who carried them, the names of the molecules they carried, the cities where their proteins were sequenced, and the color wheel of their derivatives that lights up almost every modern microscope. But none of that has happened yet. Tonight, in the late summer of 1961, the jellyfish are simply in the bucket. And the bucket is on the dock. And the dock is at Friday Harbour. And the light at Friday Harbour is going. The man who will eventually understand the green light has, on this particular evening, a notebook in his pocket, and a pencil behind his ear, and the wide, easy patience of a person who knows that nothing is going to happen quickly. He has not yet ground up his first jellyfish. He has not yet emptied his first batch of seawater into a sink. He has not yet seen the strange flash that will, in a few weeks, redirect his entire research career, and through it, through all the patient years that follow, redirect a small but important corner of the life sciences. None of that has happened. He is just standing on a dock, watching the bay go dark, with a bucket of medusae beside him and a long, quiet summer ahead. We will get to him. We will get to the night when his sink flashes blue. We will get to the calcium ions and the proteins and the gene and the worm and the color wheel and the medal. We will get to all of it, but we will get to all of it the way it actually arrived. Slowly, quietly, one bucket at a time. For now, in the cold half-light of Friday Harbor in 1961, the only thing in the world that matters is the shape in the Wrack line and the way it pulses pale green twice in the deepening dark. Nobody else sees it. The dock is empty. The water is dark. It does not matter. The pulse happens whether or not it is observed, the way the tide happens whether or not it is measured, the way light happens whether or not anyone is looking. A crystal jelly that nobody yet calls famous. That is what we left at the end of the dock in the late summer of 1961. The bucket sits patiently. The medusae drift in slow circles. Nothing happens. The tide goes out a little further. The kelp on the Wrack line dries at the edges and curls very slightly in the cooling air. We do not need to rush back to the laboratory. The jellyfish are not going anywhere. They have, in fact, never been anywhere. They have always been here, at this northern threshold of the Pacific, gently pulsing, gently feeding, gently giving off a green that nobody has yet learned to read. Let us look at them for a little while, the way they look without the laboratory. Aequorea victoria is a hydrozoan medusa. That is to say, it belongs not to the great showy class of true jellyfish, the Scyphozoa, the moon jellies, the lion's mane, the sea nettle, the giants of the salty imagination, but to a quieter, smaller, more papery class called the Hydrozoa. Hydrozoans tend to live double lives. They have a polyp stage, a small attached creature anchored to a rock or a piling, and they have a medusa stage, the floating one we recognize as a jellyfish. Aequorea victoria is one of these. Its taxonomic address is, for those who like such things, Kingdom Animalia, Phylum Cnidaria, Class Hydrozoa, Order Leptothecata, Family Aequoreidae, Genus Aequorea, Species Victoria. The genus was named in 1809 by the French zoologists François Perron and Charles-Alexandre Le Sueur, after the Latin aequor, the level surface of the sea. The species name was added by Murbach and Shearer in 1902. So by the time we reach our 1961 dock, this animal has had its formal name for almost 60 years. It has, however, been almost completely uninteresting to almost everyone. It is a small medusa. The largest specimens grow to the size of a saucer or a small dinner plate. Most of what you see in the water is much smaller than that, 3 or 4 centimeters across, sometimes less. The bell is gelatinous, rounded, and so transparent that in life you can read the shapes of fish and kelp through it as if it were a thin sheet of glass. There is a velum, a thin, muscular ring at the bell margin, which the animal uses to push water and propel itself through slow, gentle pulses. There are tentacles, uneven in number, increasing as the animal grows, up to about 150. The tentacles bear nematocysts, the small, stinging organelles that all cnidarians carry, and they help the medusa catch and subdue its prey. The stings have no effect on humans. You can lift a crystal jelly in your hand and it will not hurt you. It will, however, almost certainly fall apart. From the center of the underside of the bell rises a contractile mouth, the manubrium. The manubrium is, in some ways, the only piece of the medusa that looks even slightly threatening. It can stretch, distend, and extend in a way that would seem cartoonish if it were not so quiet. A crystal jelly can, on a good evening, swallow another animal half its own size, simply by reaching out and folding its mouth around it. From the manubrium radiate up to 100 fine canals, the radial canals, that run all the way out to the bell margin, distributing seawater and nutrients through the soft body. In larger specimens, you can see the gonads as whitish thickenings along the length of these radial canals. Each medusa is either male or female. Their reproductive cycle, like everything else about them, is unhurried. The life cycle, when you draw it out, looks almost more vegetable than animal. In late spring, on a hard surface somewhere on the bottom, a piling, a stone, a piece of submerged wood, there is a tiny hydroid colony, a few millimeters across, attached to the substrate. This colony, in response to environmental cues that nobody has yet entirely identified, begins to bud off small, free-swimming jellyfish. These tiny medusae drift up into the water column. They feed, they grow. They join the slow plankton drift through the summer. Once they reach about 3 centimeters in diameter, they begin producing eggs and sperm in their gonads, releasing them into the water in response to a daily light cue. The eggs are fertilized externally. They drift. They eventually settle. Some of them find a hard surface, attach, and become next year's hydroid colony. The medusae themselves, having finished their summer work, generally die in the autumn after about 6 months of life. The whole cycle is gentle. Nothing in it lasts very long. Nothing in it is loud. Aequorea victoria are inefficient swimmers. The biologist who measures their swimming velocity finds, somewhat surprisingly, that the bigger the animal, the more or less the same speed it goes. They do not pursue prey. They depend instead on what physical biologists call passive energy recapture, using the small amount of muscular contraction available to them to rebound off pressure differentials in the water around the bell, drifting with grace rather than urgency. They eat what comes to them, mostly soft-bodied creatures, mostly other gelatinous things, copepods sometimes, crab larvae sometimes, barnacle nauplii sometimes, other ctenophores, other appendicularians, other small hydromedusae. And, here is the strange part, sometimes other Aequorea, given the chance, swallowed half whole by the contractile mouth of a slightly larger neighbor. Cannibalism, on the rare days when conditions allow, is part of the life of the crystal jelly. This, by the way, is something the marine biologist Jennifer Purcell documented in 1991. She showed that Aequorea is not just a passive drifter, but what is called an intra-guild predator. A predator that eats other predators in the same trophic guild, competing with the very animals it dines on. Where Aequorea blooms thicken in the bay, certain populations of small zooplankton thin out almost in inverse proportion, indicating that this delicate-looking animal is, ecologically, doing more work than its size would suggest. The crystal jelly is not just a glass ornament adrift in the bay. It is a small, slow, methodical predator. It is, in its own way, an ecosystem engineer. It has its own predators in turn. The big lion's mane jelly, Cyanea capillata, will eat Aequorea when it encounters one. So will siphonophores, the colonial drifters of the open ocean. So will other ctenophores. There are documented cases of Aequorea being eaten by Aequorea. There are also small parasitic hyperiid amphipods, Hyperia medusarum in particular, that latch onto the underside of the bell, sometimes burrowing into the gelatinous tissue, occasionally living inside the gut or the radial canals like commensal travelers in the body of their host. None of these activities, intriguingly, seem to be lethal to the jellyfish. The jelly drifts on, with its parasite, with its half-digested copepod, with its quietly accumulating life history, towards autumn. A small, transparent, soft, slow, mostly invisible predator with up to 150 tentacles, up to 100 radial canals, gonads that mature daily on a light cue, a contractile mouth that can swallow half its own size meals, a 6-month adult lifespan, a fondness for cold Pacific waters, and a propensity for being eaten by things larger and more famous than itself. It has, however, one specific and remarkable feature, which is the reason we are sitting here together at all. Around the rim of the bell, in small, concentrated granules, there are 2 co-localized proteins that, between them, can produce light. We have already named them. We will name them again. The first is aequorin, a calcium-activated photoprotein. When a calcium ion enters the right pocket of aequorin, the protein changes shape, oxidizes a small luciferin called selenterosine that it has been holding within its structure, and releases the resulting energy as a flash of blue light. This blue light has a wavelength of about 465 nanometers, somewhere between deep ocean and a cold winter sky. The second is the green fluorescent protein, GFP. GFP does not, by itself, make light. It absorbs blue and ultraviolet light and re-emits that energy as green. In the granules at the rim of the bell, aequorin's blue is absorbed by the GFP. The GFP shifts the energy into the green range. And so what you see, what we saw on our Wrack line dock, is not the blue flash of aequorin alone, but the green echo of GFP, sitting between the blue and your eye. This whole arrangement, this calcium blue-green sequence, is called Förster resonance energy transfer. It is named for Theodor Förster, the German physical chemist who first described it in 1948. It is a phenomenon that turns up everywhere in modern biology, But its first, thoroughly characterized natural example is exactly here, in this small Pacific medusa, on this dock in 1961. The biological purpose of all this, the reason a crystal jelly bothers to convert its blue into green, is, even now, not entirely clear. It is one of the small, unresolved questions of marine biology. Perhaps the green light deters certain predators. Perhaps it confuses them. Perhaps it has no purpose at all and is simply a piece of chemical accident, retained because there is no strong selection pressure to lose it. We do not know. The jellyfish does. It simply continues to glow faintly around the rim when something disturbs it. This is the animal at our feet. Quiet, transparent, gelatinous, hungry, 6 months old, named after the level surface of the sea. Its body is a small carrying case for two extraordinary proteins that the rest of biology, in the late summer of 1961, has only the dimmest awareness of. To the man on the dock, it looks like work. To the world, eventually, it will look like a gift. Tonight, it just looks like a small crystal jelly drifting in a bucket, almost invisible, doing absolutely nothing dramatic, holding two ribbons of glowing protein at the rim of a bell that nobody on the planet has yet thought to take apart. The wind off the bay is light. The light is going. The medusae drift in slow circles in their bucket, doing the only thing they know how to do, which is to be themselves. We will let them rest here for a moment, in the gathering dark, and turn now to the man who, in a few weeks' time, will start carrying these creatures up to a sink, and who will discover, almost by accident, what is inside them. We will get to the man with the bucket, but first, a Sea-Firefly. A boy in Isahaya, a train ride to Nagoya, a flash of an atomic bomb, and a strange light at the rim, two ribbons of glow. We will get to the man with the bucket. But first, as promised, a Sea-Firefly. The man in question is Osamu Shimomura. He was born on the 27th of August 1928 in the small town of Fukuchiyama in the central Japanese prefecture of Kyoto. His father was an officer in the Imperial Japanese Army, and so the family moved often through the boy's early life, with stretches in Manchukuo, what was then Japanese-occupied Manchuria, and stretches in Osaka. By the time of the Pacific War, the family had settled in Isahaya in Nagasaki Prefecture, 25 kilometers from the city of Nagasaki itself. On the 9th of August 1945, the American B-29 bomber Boxcar crossed the southern coast of Japan, climbed over the Kyushu cloud layer, and dropped a plutonium implosion device on the city of Nagasaki. Shimomura, then 16 years old, was in the family home in Isahaya, well outside the immediate blast zone, but well within the reach of the flash. He recalled later that he heard the bomber overhead before he saw the explosion. The brightness of the detonation, even at 25 kilometers, was enough to blind him for about 30 seconds. Some hours later, he was caught in the dark, sticky fallout that the survivors of those bombings would call kuroi ame, black rain, radioactive moisture mixed with soot and ash, settling slowly out of a torn sky. He survived. The atomic bomb did not kill him. The flash did not permanently blind him. The black rain did not, as it killed many people who walked through it, kill him. He would live another 73 years, and he would carry the memory of those minutes very quietly with him for all of them. The Japan he came of age in was not the Japan of unlimited educational opportunity. The Nagasaki Medical College had been completely destroyed by the bomb. Its pharmacy school, the College of Pharmaceutical Sciences, had been forced to relocate to a temporary campus near Shimomura's home in Isahaya. He has said, plainly and without ornament, that he had no particular interest in the subject. He went to pharmacy school because pharmacy school was what was nearby. There was nothing else to enroll in. He earned a Bachelor of Pharmacy from Nagasaki Medical College in 1951. He stayed on as a lab assistant for 4 more years. He had not yet found his subject. He was simply waiting, patiently, the way patient people. What came next was an introduction. In 1956, his mentor at Nagasaki helped him find a position as an assistant to a professor named Yoshimasa Hirata at Nagoya University, several hundred kilometers east. Hirata was a quiet, demanding, brilliant organic chemist. He gave Shimomura almost immediately a problem that no one had been able to crack. The problem was a small crustacean. The Japanese name for this animal is Umi Hotaru, Sea-Firefly. Its scientific name is Vargula hilgendorfi. It is a tiny ostracod crustacean, no more than a few millimeters long, that lives in the coastal waters around Japan. When you crush its remains and add water, the moistened powder produces a bright, brief, blue glow. This was the puzzle Hirata gave the new assistant. Why does the sea-firefly glow? What chemical is responsible? Identify it. Purify it. Tell me what its structure is. The assignment turned out to be enormously difficult. Shimomura spent years on it. He learned to weigh tiny quantities of luminescent material, to crystallize them, to recrystallize them, to chase a chemical answer through extracts that would inactivate the moment they came into contact with the wrong solvent. He developed, in the process, a kind of patience that the wider scientific world would later find astonishing. He was capable of working with 2.5 tons of dried sea-firefly material to obtain less than half a gram of a purified compound. He would, in his career, do similar things repeatedly. It was not because he had a special love of suffering, it was because the problem required it. In 1957, with Hirata's guidance, he succeeded. He crystallized the small molecule responsible for the sea-firefly's glow. He named it Cypridina luciferin, after the older name of the genus. He published his preliminary findings in a paper in the Bulletin of the Chemical Society of Japan, titled simply Crystalline Cypridina luciferin. He earned his Master of Science in Organic Chemistry in 1958. He earned his doctorate in 1960. This paper, this small, careful piece of work on a small, careful problem, far from the centers of biological glamour, was the thing that changed his life. The paper made its way by some mailroom route to the desk of a biologist at Princeton University named Frank Johnson. Johnson was an Americanist of the old school, courteous, hard-working. He had been studying the chemistry of glowing animals for decades. He read Shimomura's paper. He recognized the work as the work of someone who could do what he himself could not. And he wrote. In 1960, Shimomura accepted a position with Johnson at Princeton. He arrived in the United States with a Japanese pharmaceutical degree, a doctoral thesis on a sea-firefly, almost no English, and the same patience he had been carrying around since the war. Johnson met him at the train. Shimomura recalled later that he was somewhat puzzled by what came next. Johnson did not want him to keep working on Cypridina. Johnson wanted him to work on a jellyfish. The jellyfish was Aequorea. Some accounts give it as Aequorea, the Atlantic and Mediterranean form. Some accounts give it as Aequorea Victoria, the Pacific form. Shimomura himself, by the end of his career, considered them synonymous, though taxonomists have argued about the boundary for decades. What mattered was that Aequorea, like Cypridina, glowed, and that nobody had yet purified the molecule responsible. Johnson had been trying. Johnson had not succeeded. Johnson wanted Shimomura's hands, and so, beginning in the summer of 1961, Osamu Shimomura began the strange, long pilgrimage that would define the rest of his career. Each summer, he would travel from Princeton, New Jersey, all the way across the continent to the small wooden field station at Friday Harbor on San Juan Island, off the northwestern corner of Washington state. There, with Johnson, with various students and assistants, with his wife Akemi, herself an organic chemist and his lifelong scientific partner, he would collect Aequorea jellyfish from the docks of the harbor in nets and buckets, hour after hour, evening after evening, summer after summer. By his own count, between 1961 and 1988, he collected approximately 1 million crystal jellyfish from the waters around Friday Harbor. The number is not poetic, it is a count. He had a notebook, he kept it. For each summer's worth of jellyfish, he and his collaborators would cut off the rims of the bells, the part that contained the glowing granules, and feed those rims through grinders, through filters, through columns of separation media. The work was tedious to a degree that defies casual description. The protein they were chasing was unstable and inactivated easily and could be lost forever in a single careless step. In the summer of 1961 and through the year that followed, however, he made a breakthrough. The story, told later in his 1995 reminiscence in the Biological Bulletin, has the small, wonderful texture that the best laboratory stories often have. He had, after weeks of work, a small purified extract of the protein from the rims of Aequorea bells. He poured it down a sink at the end of the day, the way you pour out a solution that is not behaving the way you wanted it to. The sink, however, had not been completely cleaned. There was still in the bottom a small amount of seawater from earlier in the day. The instant the extract met the seawater, the entire sink lit up. A bright, brief, intense blue light, lasting only a few seconds, then fading. It was, in his words, a startling moment. The protein, which had been giving him no light at all when he tried to provoke it on his bench, suddenly glowed when it met seawater. It did not take him long to work out why. Seawater contains, among other things, calcium ions. The protein was responding to the calcium, and so, with patient subsequent work, he and Johnson established that the photoprotein from Aequorea required calcium to glow. They named the protein Aequorin. They published the discovery in 1962. The paper, by 21st century standards, is brief. It is also, by 21st century standards, foundational. Aequorin became the first widely used calcium reporter in cell biology. By 1967, the physiologists Ridgway and Ashley were micro-injecting it into single muscle fibers of barnacles and watching the protein flash blue every time the muscle contracted. They were watching, for the first time in the history of biology, calcium signals inside a living animal. But Shimomura, even in that founding paper, had noticed something else. There was, he wrote in his 1995 reminiscence, in the original purification, a small protein that was not Aequorin. It was a slightly different size, a slightly different color. It did not flash blue when calcium hit it. It glowed faintly green when you shone ultraviolet light on it. He set it aside. He kept it in a jar. He named it the green protein. Eventually, somebody else would call it the green fluorescent protein. Eventually, that small green glowing thing in a side jar would change the way every microscopist on Earth did their work. That, however, lay in the future. For the moment, in 1962, on a wooden dock in Friday Harbor on the cold Pacific shelf of North America, the thing that mattered was that a sink had flashed blue when it should not have, and that a Japanese organic chemist with a Sea-Firefly thesis and a deep memory of the Nagasaki bomb had been patient enough to ask why. There would, of course, be many more sinks, many more buckets, many more summers. Shimomura's wife, Akemi, herself an organic chemist, was at his side through most of it, washing glassware, weighing extracts, keeping the record straight. The Friday Harbor field station lent them space. The marine biologists there became long-term collaborators of the project. The work, year by year, refined. The protocols got slightly cleaner, the yields got slightly higher. The buckets slowly accumulated. By the late 1960s, Aequorea was well characterized, its calcium trigger mechanism understood, its sequence partially worked out. The green protein was sitting very patiently on a shelf in a small jar. Nobody yet had a use for it. Nobody yet had even tried particularly hard to clone it. The Sea-Firefly boy from Isahaya by the late 60s was a respected American-based organic chemist with 2 important glowing molecules to his name and a third one, quietly remarkable, quietly waiting, in a labeled jar in a freezer in New Jersey. It was a sink that should not. To understand why a sink could flash blue, we first need to understand a calcium ion. Calcium is one of the most abundant metals in the human body. It accounts for about 1.5 kilograms of the average adult, most of it locked into the matrix of bone. But calcium also exists, in much smaller quantities, as a free, positively charged ion drifting in solution, calcium 2+, written in chemistry shorthand as Ca2+. In seawater, this ion is everywhere. It is one of the major dissolved cations in any ocean, anywhere on Earth, and the open Pacific is no exception. Inside the cells of almost every animal, however, calcium is much rarer. Cells go to enormous trouble to keep their internal calcium concentration low. There are pumps in the cell membrane that constantly push calcium out. There are intracellular stores, the smooth endoplasmic reticulum in particular, that sequester calcium away from the cytoplasm. The result is that, at rest, the inside of a cell typically has about 10,000 times less free calcium than the outside. The gradient is enormous. The biology that depends on it is enormous too. Because when a cell decides to do something, fire a nerve signal, contract a muscle fiber, release a hormone, secrete a digestive enzyme, divide, one of the very first things that happens, almost universally across the animal kingdom, is that calcium ions are allowed to flood briefly into the cytoplasm from the outside or from the internal stores. The calcium signal is, in many ways, the master signal of biology. It is the on switch. It is the trigger. It is what muscle uses to contract. It is what neurons use to release neurotransmitter. It is what an egg uses to recognize that it has been fertilized. It is what the heart uses to beat. In 1961, when Osamu Shimomura began grinding up jellyfish at Friday Harbour, the importance of calcium signaling was beginning to be appreciated, but there was almost no good way to watch a calcium signal happen. Cells were small. Calcium ions were invisible. The fluxes were brief. You could measure the average calcium content of a piece of tissue, but you could not, in any practical sense, watch calcium move through a single living cell in real time. This is the world into which a glowing protein from a Pacific medusa was about to fall. The world that desperately needed something that could turn an invisible calcium signal. The protein Shimomura purified in the small back room at Princeton and the rough laboratory at Friday Harbour did exactly that. He named it Aequorin, after the genus Aequorea. It is a small protein. The polypeptide chain alone is about 21 kilodaltons in mass, which is a way of saying it is a relatively short protein, on the order of 190 amino acids. But the working Aequorin molecule is not just polypeptide. It carries within it a small organic molecule, a luciferin called silenterazine, bound in a particular oxidized form known as silenterazine 2-hydroperoxide. This complex, protein plus preloaded silenterazine, is what we mean when we say the word Aequorin. Within the protein, three particular structural motifs, called EF hands, provide binding pockets for calcium ions. EF hands are an evolutionarily ancient calcium binding fold. They appear in calmodulin, in troponin C, in myosin light chains, in dozens of other regulatory proteins across animal life. Aequorin happens to carry three of them. Two of those EF hands have an extremely high affinity for calcium, about 20 times greater than the third. Studies show that all three sites are functional, but only two need to bind calcium for the protein to fire. When those two calcium ions bind, the protein changes shape. The conformational change brings the bound silenterazine 2-hydroperoxide into a configuration where the molecule can decompose. It splits, releasing carbon dioxide and producing an excited form of a different small molecule called silenteramide. As the excited silenteramide returns to its ground state, it releases the energy difference as a single photon, a photon of blue light with a wavelength of about 465 nanometers. To the eye, in the dark, that light is a soft, cool blue, a color somewhere between deep ocean and the cold half-light of a January dawn. The whole reaction is, in a sense, a chemistry lesson in microscopic form. Calcium binds, the protein folds. The peroxide on the silenterazine breaks. The molecule loses carbon dioxide. The remaining piece is briefly excited. The excited piece relaxes. The relaxation releases blue light. The whole thing happens in less than a second. This is what Shimomura saw in the sink in 1961. The seawater that had been left behind contained calcium. His purified extract contained Aequorin with its The instant the two met, all the Aequorin in the sink fired at once. The basin of the sink lit up blue. He recognized the color. He recognized that it was the same color the live jellyfish flashed in the dark when you disturbed them. And he recognized almost immediately that calcium had to be the trigger. There was no other plausible candidate in the seawater. The consequences of this realization extended well beyond the jellyfish. Within a few years, biologists had learned that aquarin could be loaded into living cells and that, when they made it, the protein would fire when the cells released calcium. In 1967, the physiologists D.A. Ridgway and Christopher Ashley took aquarin and micro-injected it into the single giant muscle fibers of barnacles. Barnacles, when they eat, contract their muscle fibers in slow, steady rhythms. Ridgway and Ashley watched the aquarin in those fibers flash blue with each contraction. It was the first time anyone had watched a calcium signal happen in real time inside a living animal. They published the result. The result was foundational. For the next 20 years, before Roger Tsien's small molecule indicators took over the cell biology calcium market, aquarin was the workhorse calcium reporter. People injected it into squid axons. They injected it into mammalian cells. They expressed it, eventually, recombinantly by cloning the gene. They fused it to other proteins. They used it to study cardiac muscle, plant cells, neurons, mitochondria. The small Pacific jellyfish, almost without anyone noticing, had given biology one of its first universal tools for watching the master signal of all signals. Aquarin is, by laboratory standards, robust. The protein survives, with most of its activity intact, even after 2 minutes of heating at 95 degrees Celsius. Treatment with 6 molar urea or 4 molar guanidinium chloride, both common protein denaturants, does not destroy it. Soilenterazine, the small molecule that does the actual chemistry, is hydrophobic enough to cross many cell membranes on its own, which means that even today, you can express the apoaquarin protein in cells of plants, fungi, mammals, or insects, then add soilenterazine to the medium and have working aquarin inside the cell within minutes. This trick, adding the small molecule from outside, having it cross the membrane and recombine with the protein, soilenterazine itself was not for many years identified. It is a small heterocyclic compound, an imidazopyrazinone, that turns out to be the most widespread luciferin in the open ocean. The same molecule, with small variations, drives the bioluminescence of deep-sea jellyfish, of certain shrimp, of the bony fish family Vincigueria, of the radiolarian Thalassicola. It is the marine luciferin. It was identified in 1977 by a team of researchers, building in part on Shimomura's earlier work, finally giving the small marine luciferin a face and a structure. The soilenterazine that Aequorea uses is the same soilenterazine that hundreds of other marine organisms use. The protein that holds it is what makes Aequorea distinctive. Aquarin is, in other words, a particular calcium-triggered packaging of a luciferin that the ocean shares with many of its other glowing creatures. The Medusa did not invent the chemistry of light. It simply learned, somewhere in its evolutionary past, how to gate that chemistry on calcium, and how to keep the resulting blue flash where the eye and predator could see it, at the rim of the bell, in small, bright granules, ready to fire. This last detail, that aquarin lives at the rim in concentrated granules, was first noted with characteristic care by E. Newton Harvey, an American biologist who had been studying Aequorea bioluminescence since 1921, 4 decades before Shimomura arrived. Harvey could not isolate the protein. He had no purification methods of the kind that Shimomura would develop. What he could do was watch a jellyfish glow in the dark and report accurately that the glow appeared as a ring around the bell and, strangely, that the glow continued even when the animal was deprived of oxygen. This second observation puzzled him for years. Most known bioluminescent reactions required molecular oxygen. The Aequorea reaction did not seem to. We know now what Harvey could not. The reason Aequorin can fire even in the absence of oxygen is that the oxygen has already been used. The protein stores its prosthetic group in the pre-oxidized form selenterrazine 2-hydroperoxide. The peroxide bond holds the energy. Calcium just opens the door. The blue flash that you see when Aequorea is disturbed is, in chemistry terms, the spending of an energy. By the late 1960s then, calcium and Aequorin were beginning to be tied together in the literature. The protein had been published. The discovery was Shimomura's and Frank Johnson's. The chemistry had been worked out. The applications were beginning to multiply. In a textbook of cell biology from any year after about 1970, Aequorin would be mentioned somewhere, usually a small paragraph in the chapter on cellular signaling, alongside calcium-induced calcium release and the contraction of the sarcomere. The protein had become a tool. But all the way through this period, in the rim of the bell of every living Aequorea, there was a second protein, smaller, quieter. Not a photoprotein in the strict sense, not a luciferase, not a calcium reporter. It made no light on its own. It simply absorbed blue light and re-emitted it as green. Shimomura had noticed it. Shimomura had set it aside in a small jar. Shimomura had named it the green protein. He had even, in a paper in 1974, with William Maurice and Frank Johnson, described how it absorbed the blue energy from Aequorin and shifted it into the green range. The result was elegant. The result was published. The result, however, did not, for almost 2 decades after that paper, find a use. It was simply too obscure. It was a curiosity. It belonged to a single species of soft-bodied medusa in a single corner of the Pacific, and it required, for its blue energy source, a calcium-triggered photoprotein that itself required laborious purification from millions of jellyfish. Nobody in 1974 knew yet that the green protein could be cloned, expressed in any cell, and made to glow on its own. Nobody in 1974 knew yet what was hiding inside its barrel. The world had Aequorin. The world had calcium. The world had the blue flash. And behind the blue, there was always, somewhere in the rim of the bell, a green. Where did the green come from? It came from inside a barrel. This is, structurally, the strangest and most elegant fact about the green fluorescent protein. The molecule that converts an Aequorea jellyfish's blue light into green is shaped, almost exactly, like a small can. A barrel, a cylinder, a closed pocket of protein with one chemical machine running through the center and the rest of the molecule wrapped around that machine like a wall. Let us look at it for a while. Tonight, we have time. The full GFP molecule is a chain of 238 amino acids. That is, by protein standards, fairly small. Many enzymes are 5 times longer. Many structural proteins are 20 times longer. GFP is compact. It folds into a single, self-contained structural unit, which biochemists call a domain. There is no second domain. There are no flexible tails. There are no flopping arms. The whole protein folds into one shape, and that shape is the barrel. The barrel is built from 11 beta strands. A beta strand is a section of polypeptide that lies in a flat, extended configuration, the carbonyl groups of the backbone pointing one direction and the amide hydrogens pointing the other. Beta strands like to find each other. They like to lie down side by side, hydrogen bonding across the gap between them, building flat sheets. These sheets are the structural backbone of an enormous fraction of protein architecture. Some proteins use them flat, like a slab. Some proteins curl them. GFP curls them. The 11 beta strands of GFP wrap around in a cylinder, hydrogen bonding to each other in sequence until the 11th strand bonds back to the first, and the whole cylinder closes. The resulting structure is a nearly perfect cylinder, about 42 angstroms long and 24 angstroms in diameter. An angstrom is a tenth of a nanometer, 10 billion to the meter, a small enough unit that almost any object you have ever seen contains an unimaginable number of them. The barrel, in human scale terms, is laughably small. In molecular terms, it is roomy. It is a cathedral. At the top and the bottom of the cylinder, 5 short alpha helices form caps, sealing the barrel at its ends. The wall is the 11 beta strands. The caps are the helices. The interior is, as far as proteins go, watertight. Solvent molecules, water in particular, are largely excluded from the inside of most proteins is dry, but only a few have such a complete and reliable seal as GFP. The structure is so distinctive that biologists gave it its own name, the beta can. The shape of the can is unique to the GFP family. And what then sits inside the can? A single small chemical machine threaded through the center of the barrel along an internal alpha helix that runs the long axis of the structure. The machine is the chromophore. It is the part that actually fluoresces. It is, depending on how you count, 3 amino acids in size, fused into a small ring that did not exist when the protein was first translated. The chromophore is a molecule called 4-p-hydroxybenzylidene imidazolidine 5,1, abbreviated HBI. The name is mechanical. The molecule, in chemistry terms, is a hydroxybenzylidene imidazolinone, a planar, conjugated aromatic system that absorbs blue and ultraviolet light and re-emits in the green. HBI is what does the work. HBI is what flashes when blue light arrives. HBI is, in the strict optical sense, the green fluorescent protein. The miraculous thing, the thing that makes GFP unlike any other glowing molecule that biology had previously stumbled across, is that the cell does not synthesize HBI. Not directly. Not at all. The cell synthesizes the polypeptide chain, the long ribbon of 238 amino acids with the standard amino acid sequence at positions 65, 66, and 67 of serine, tyrosine, glycine. And then, once the polypeptide is folded into the barrel, those 3 amino acids, serine 65, tyrosine 66, glycine 67, undergo a remarkable spontaneous chemical reaction inside the protein with no help from any other enzyme. They cyclize, they dehydrate, they oxidize, they become HBI. This process is called maturation. It happens inside the folded barrel, in the dark, automatically, given only the presence of molecular oxygen. The cell does not provide a special enzyme to drive the reaction. The protein drives it itself. The barrel itself is the catalyst. The folded structure brings the 3 amino acids into a precise geometry, and certain neighboring residues, glutamate 222, arginine 96, glutamine 94, histidine 148, stabilize the intermediates, donate hydrogens. The result, after about half an hour of patient internal chemistry, is a fully formed HBI fluorophore, locked at the center of the barrel. It is, in the literal sense of the word, autocatalysis. The protein assembles its own working part. The cell does not need to provide cofactors specific to the jellyfish. There are no exotic ingredients. The reaction needs only oxygen, which any cell that respires has plenty of, and a few hours of patience. Once the chromophore is formed, it cannot easily come apart. The reaction is essentially irreversible. The molecule glows, and the barrel keeps it glowing. The 11-stranded wall of the can, with its dry interior, protects the chromophore from the one thing that would otherwise quench its fluorescence: water. A free HBI molecule in solution, away from a barrel, glows extremely faintly. Most of its excited state energy is lost to vibrational coupling with the surrounding water before it can be re-emitted as green light. Inside the barrel, however, water is kept out. The chromophore can relax only by emitting a green photon. The barrel is, in effect, an isolation chamber for one of the simplest and most efficient light-emitting tricks in chemistry. This robustness extends well beyond water exclusion. Wild-type GFP is, by protein standards, almost absurdly tough. It survives exposure to detergents that would dissolve other proteins. It resists most proteases. It tolerates treatment with guanidinium chloride, a chemical that unfolds nearly every other globular protein in the cell. It shrugs off temperature changes that would destroy a less compact molecule. It can be boiled briefly and still recover its fluorescence on cooling. Its half-life inside a cell is hours rather than minutes. The 2 parameters that biophysicists use to characterize a fluorophore are extinction coefficient and quantum yield. The extinction coefficient measures how strongly a molecule absorbs light at its peak wavelength. The quantum yield measures how often, having absorbed a photon, the molecule actually re-emits as a fluorescent photon rather than losing the energy as heat. Wild-type GFP has an excitation peak around 395 nanometers in the ultraviolet, and a smaller secondary peak around 475 nanometers in the blue, with an emission peak at about 509 nanometers in the bright green. It is, in other words, a molecule that absorbs blue light efficiently and turns most of what it absorbs into green light. The 2 excitation peaks turn out to be, in their own way, a fingerprint of how the chromophore lives inside the barrel. Serine 65, the first of the 3 chromophore residues, can donate a hydrogen to glutamate 222, depending on the exact protonation state of the system. The protonated and deprotonated forms of the chromophore have slightly different absorption spectra. Wild-type GFP, with its native serine, oscillates between the 2 forms in solution, giving the dual peaks. Roger Tsien would later notice this and exploit it. For now, in the late 1970s and early 1980s, no one knows any of this in detail. The crystal structure of GFP would not be solved until 1996. The autocatalytic mechanism would not be fully understood until later still. The biophysicists who would decode the molecule are, in the late 70s, undergraduates and graduate students. The relevant technologies, recombinant expression, X-ray crystallography of small soluble proteins, site-directed mutagenesis, are either young or not yet practical. The molecule is, for now, a curiosity in a side jar in Frank Johnson's freezer at Princeton, with a few notes in Shimomura's notebook describing how it had been purified, how it absorbed and emitted, and how it shifted Aequorin's blue into green. A single, small protein. 238 amino acids, 11 beta strands, 5 short helices. One internal chromophore that builds itself out of 3 amino acids and a breath of oxygen. A quantum yield of 7/10. A green emission at 509 nanometers. A handful of stabilizing residues. A perfectly closed cylinder. This was, structurally speaking, the answer. Inside the beta can was the green. The barrel kept the chromophore dry. The chromophore released a photon every time blue light arrived. The dry interior was the secret. The barrel was the secret. The protein, if you could only get it out of the jellyfish and into another organism, would carry its own little green light with it wherever it went, automatically, with no help, with no luciferin, with no enzyme. It only needed oxygen. It only needed, in other words, what almost every cell on Earth already has. The biological purpose of GFP in the jellyfish remained. The animal could fire blue with Aequorin alone if it wished to. The conversion of blue to green by GFP is, evolutionarily, a small refinement. Perhaps it shifts the color into a wavelength better matched to the visual systems of certain predators. Perhaps it serves to distract or confuse. Perhaps it is simply a chemical accident, retained because there is no strong selection against it. We do not know. The Medusa does not, again, care that we do not know. It glows green, faintly, at the rim when something disturbs it, the way it has been doing for tens of millions of years. What the late 1970s cell biologist would have given, however, to be able to take that small barrel out of the jellyfish, attach it to any protein of interest, express it in any cell, and watch the cell glow exactly where that protein was. The dream was obvious. The technology was almost there. The only piece missing was the gene. The gene was, by 1978 or so, theoretically isolable. Recombinant DNA techniques had been developed. The cloning of small messages from rare proteins was beginning to be done routinely in some laboratories. Someone, somewhere, would eventually try to pull the GFP gene out of an Aequorea cDNA library and turn it into a transferable tool. That someone was Douglas Prasher. He was a biochemist working at the Woods Hole Oceanographic Institution in Massachusetts. He had read Shimomura's papers. He had read the photophysics of the protein. He understood what GFP could become, if only it could be cloned. He set about, in the 1980s, doing exactly that work. But the gene for that small barrel was, in the late 1970s, still on a shelf in a freezer in not a small molecule, not a dye, not a stain, a gene. This is the central pivot of our story. The reason GFP became the most important protein in the history of cell biology is not that it glows. Many things glow. Phosphorescent zinc sulfide glows. Fluorescent glows. Quinine in tonic water glows under ultraviolet light. The reason GFP changed everything is that the entire glowing apparatus is contained in a single sequence of DNA, with no requirement for any small molecule cofactor that the host cell has to provide. Move the gene into a worm, a fly, a fish, a plant, a bacterium, a mammalian cell, and the cell will, on its own, with no further chemistry required, build a glowing protein. That is the trick. That is what makes the whole story possible. But to make that trick available, somebody first had to find the gene. The gene was, in the late 1980s, still uncloned. Equorin had been cloned separately by several groups in the early 80s. GFP had not. A pair of scientific obstacles slowed the work. The protein was less abundant than equorin. The amino acid sequencing methods of the early 80s were not yet quite good enough to derive a clean sequence from the small amounts of GFP that could be extracted from the rims of jellyfish bells. And the funding for such an obscure project was thin. Pacific Medusa proteins were not, at the time, considered a likely investment. The person who solved this problem was Douglas Prasher. Prasher was a biochemist at the Woods Hole Oceanographic Institution in Massachusetts. He had read the GFP literature carefully. He understood in detail what the protein could become if it could be cloned. He spent the better part of the 1980s chasing the GFP gene through Aequorea cDNA libraries against the resistance of partially degenerate amino acid sequence data, against funding cycles that turned uncertain, against the small, slow doubts that any scientist working on an obscure marine protein in those years had to confront. In 1992, Prasher succeeded. He published the full primary structure of the gene encoding wild-type GFP in a paper in the journal Gene, titled Primary Structure of the Aequorea victoria Green Fluorescent Protein. The paper gave the full nucleotide sequence. It gave the inferred amino acid sequence. It described how the gene had been recovered. It made the gene available. This was the moment the field could have, finally, taken off. It did not. Prasher's funding was running low. He could not, in his own laboratory, afford to do the experiments that would prove what GFP could become as a cellular tool. He did, however, have the cDNA, the cloned working copy of the gene. He did the most generous thing a scientist can do when he runs out of resources to pursue his own work. He sent the cDNA, free of charge, to colleagues who he thought might be able to take the next step. One of those colleagues was Martin Chalfie, a neurobiologist at Columbia University in New York. Chalfie had been born in Chicago in 1947, had earned his PhD in neurobiology from Harvard, and had spent most of his independent career studying the small soil nematode Caenorhabditis elegans, a transparent worm about 1 millimeter long with exactly 959 cells in its adult body, every single one of which had been mapped, named, and lineage traced by the painstaking work of Sidney Brenner, John Sulston, and Robert Horvitz. C. elegans was a biologist's dream organism for one specific reason. It was transparent. You could put a worm under a microscope and watch its cells in real time in any tissue. The animal cooperated. Chalfie had been studying touch sensation in C. elegans. The neurons that mediate touch were known by name, by lineage, by connection. What Chalfie wanted was a way to mark them in the living animal so that he could watch them respond to mechanical stimulation under the microscope. The traditional histological dyes required killing the animal and fixing the tissue. The Aequorean route required micro-injecting protein into individual cells, a technique that did not scale. What Chalfie needed was a genetically encoded marker, a gene that, when expressed, would make a cell visible without killing it. In the late 1980s, Chalfie heard a seminar by the biologist Paul Brem about jellyfish bioluminescence and the green fluorescent protein. He recognized almost instantly what he was hearing. He looked up Prasher. Prasher, by then, had cloned the gene. Prasher sent Chalfie the cDNA. Chalfie's lab, working with a graduate student named Yuan Tu, with a postdoctoral researcher named Gia Euskirchen, and with William Ward of Rutgers, who had purified GFP for many years, started to test whether the cloned gene would work. They expressed the GFP coding sequence, with the first few amino acids deleted, for technical reasons of cloning, in heterologous cells of E. coli. The bacteria glowed green under blue light. They expressed it in C. elegans under a touch neuron-specific promoter. The transgenic worms had glowing touch neurons visible in the living animal on the bench, with no exogenous chemistry required. The protein folded, the chromophore matured, the cells lit up. This was the experiment that broke the field open. Chalfie and his colleagues published the result in the journal Science in February 1994 in a short paper titled "Green Fluorescent Protein as a Marker for Gene Expression." The paper has, in the 3 decades since, been cited more than 6,700 times. It is one of the most influential cell biology papers of the late 20th century. The reason is simple. The paper showed definitively that GFP folded and fluoresced in foreign cells without any cofactor unique to the jellyfish. The barrel was self-contained. The chromophore matured on its own. Any cell that could build the protein would build a glowing cell. The dream that had been hovering over the field since the 1970s was, suddenly, executable. 1 month later, in March 1994, Frederick Tsuji's laboratory in Japan independently reported recombinant expression of Aequorea GFP, confirming the result on a different continent. The protein worked. It was now, suddenly, a tool. In the immediate years that followed, every laboratory that worked on cell biology, on developmental biology, on neuroscience, on plant biology, began to think about how to use GFP. The advantages were obvious. You could tag a gene of interest with the GFP coding sequence, drop it into a cell or an organism, and the cell would, from then on, build the target protein with a glowing tag attached. You could watch the protein move through the cell. You could watch the cell move through the tissue. You could watch developmental processes unfold in real time in living animals without killing them or fixing them. The technique, in its simplest form, did not require any specialized reagents beyond ordinary blue light and the microscope camera. The original wild-type GFP, however, had a few inconveniences.
it folded poorly at 37 degrees Celsius.
Its excitation spectrum had two peaks, neither of which matched the standard fluorescent filter sets that most fluorescence microscopes already came equipped with. Its overall brightness was modest. Its fluorescence was sensitive to pH and to chloride ions in the medium. It worked, but it was, by the standards that protein engineers were about to bring to bear on it, only an early prototype. This is where Roger Tsien arrives in our story. Tsien was, by the early 1990s, an established expert in the design of fluorescent reporter molecules. He had earned his PhD at Cambridge in 1977 with a thesis titled "The Design and Use of Organic Chemical Tools in Cellular Physiology." He had moved to Berkeley in 1982, then to UC San Diego in 1989. Throughout the 1980s, he had developed a series of small molecule calcium indicators: Fura-2 in 1985, Indo-1 shortly after, Fluoro-3 in 1989. These dyes had become the workhorse calcium reporters of cell biology. Aequorea had been the first calcium reporter. Tsien's small molecules had largely displaced it. But Tsien was, philosophically, more interested in genetically encoded indicators than in dyes. A small molecule dye had to be loaded into cells, often imperfectly, often with side effects. A genetically encoded reporter could be expressed cleanly in any cell. Tsien had been thinking since at least 1990 about how one might engineer a fluorescent protein into a useful indicator. When Chalfie published his 1994 science paper, Tsien recognized immediately that this was the chassis he had been waiting for. Within a year, Tsien's group, working with Roger Tsien and Andrew Cubitt as co-authors, reported a single-point mutation in the GFP gene that dramatically improved the protein. They published in Nature in 1995. The mutation was simple, a substitution of threonine for serine at amino acid position 65, abbreviated S65T. The effect was profound. The mutation collapsed the dual excitation peaks of wild-type GFP into a single peak at 488 nanometers. It increased the protein's overall brightness several-fold. And, perhaps most importantly for the eventual ubiquity of the tool, the new excitation peak matched, almost exactly, the spectral characteristics of FITC, fluorescein isothiocyanate, the most common small molecule green fluorophore in cell biology. Every fluorescence microscope on the planet already had a filter set for FITC. Now, those filter sets worked perfectly for GFP. In that same year, two other laboratories, independently and almost simultaneously, reported a separate point mutation, F64L, that improved the folding efficiency of GFP at 37 degrees Celsius. Combined with S65T, this gave a variant called enhanced GFP, or EGFP. EGFP was the workhorse. It folded at body temperature. It was bright. It used standard microscope filters. It is still, 3 decades later, the most commonly used GFP variant in mammalian cell biology. The quantitative numbers, for those who like them, are these. EGFP has an extinction coefficient of 55,000 inverse molar inverse centimeters and a quantum yield of 0.6. Multiplied together, the relative brightness, the practical figure of merit for a fluorophore, is 33,000 inverse molar inverse centimeters. By the standards of cell biology in 1995, this was, suddenly, a magnificent tool. In 1996, 2 crystal structures of GFP were solved within weeks of each other. The Remington Group at Oregon State University, working with the S65T mutant, published in Science. 1 month later, the Phillips Group, working with wild-type, published in Nature Biotechnology. Together, the 2 structures revealed the 11-stranded beta barrel that nobody had previously been able to see directly. The internal HBI chromophore, the supporting residues, the alpha helix running through the center, the closed caps at the ends. The structural picture became, suddenly, complete. With the structure in hand, protein engineers could finally see what they were working on. The chromophore was no longer a black box. The supporting residues were named. The hydrogen bonds were measurable. The mutations that would shift color, change brightness, or modify pH sensitivity could be designed rationally. The age of fluorescent protein engineering, which had begun cautiously with Tsien's S65T mutation, became, suddenly, an explosion. And a single letter, S65T. 65T was the first letter. The rest of the alphabet followed quickly. In the years between 1995 and roughly 2004, Roger Tsien's laboratory at UC San Diego, together with a network of collaborators and competitors at other institutions, set about engineering, systematically, the entire color wheel of GFP variants. The result was one of the most beautiful and useful campaigns of protein engineering in the history of cell biology. By the early 2000s, the small barrel from a single Pacific medusa had been turned, by a combination of rational design and directed evolution, into a palette of fluorescent reporters that covered, in slowly tightening steps, almost the entire visible spectrum. Let us walk through it the slow way. Tonight, we have time for the color wheel. The first variants were color shifts of the original GFP scaffold. The chromophore, the small HBI molecule sitting at the center of the barrel, has, as we have already noted, 3 amino acid components: serine 65, tyrosine 66, and glycine 67. Mutations at these positions, and at a small number of supporting residues nearby, change the electronic structure of the chromophore. Different electronic structures absorb and emit at different wavelengths. Different wavelengths give different colors. Mutate tyrosine 66 to histidine, and the chromophore becomes a smaller aromatic system. The absorption shifts into the ultraviolet, and the emission into the blue. Add a second mutation, tyrosine 145 to phenylalanine, to stabilize the modified chromophore, and the variant becomes blue fluorescent protein, BFP. The emission peak is around 448 nanometers. It is a deep, soft blue. It is, in 1995, the first GFP variant that does not actually emit green. Mutate tyrosine 66 to tryptophan instead, and the chromophore acquires an indole ring rather than a phenol ring. The bulkier indole forces some compensatory mutations elsewhere in the barrel to make room, but the resulting protein is brighter, slightly red-shifted from the BFP, and emits at around 476 nanometers. This variant is cyan fluorescent protein, CFP. It glows the color of soft turquoise. Improved versions, including cerulean in 2004 and M turquoise 2 in 2012, would push the cyan. The M turquoise 2 variant has a quantum yield of 0.93, which is to say that almost every photon it absorbs is re-emitted with almost no thermal loss. It is one of the brightest single fluorescent proteins ever engineered. Mutate threonine 203 to tyrosine, on the other hand, and the resulting tyrosine residue stacks against the chromophore through pi-electron interactions, lowering the emission energy. The emission shifts towards yellow. The variant is yellow fluorescent protein, YFP, with derivatives such as citrine, Venus, and Y-PET. The emission peak is around 527 nanometers. It is a warm, slightly greenish gold. With the cyan and yellow variants, biologists discovered that the two could be paired. CFP excitation overlapped with YFP absorption, which meant that when CFP and YFP were brought close enough together, within a few nanometers of each other, energy could pass directly from one to the other through the same Förster resonance energy transfer that the original Aequorea bell uses to convert blue into green. The CFP-YFP fret pair became, by the early 2000s, the most common engineered fret system in cell biology. Researchers built sensors for calcium, for cyclic AMP, for protein phosphorylation, for protein-protein interactions, all by linking a CFP and a YFP through some sensing element that would change its conformation in response to the cellular signal. The signal would change the fret efficiency. The microscope would see the color shift. The cell's internal chemistry suddenly became visible in real time. One particularly elegant example was the chameleon sensor, designed by Atsushi Miyawaki, working in Tsien's laboratory in the late 1990s. Miyawaki's design fused CFP and YFP through calmodulin, the small calcium-binding protein, plus a peptide that calmodulin binds when calcium loads it. When calcium arrived, calmodulin grabbed its peptide, the linker folded, the CFP and YFP came close, and the fret ratio shifted. When calcium left, the structure relaxed, the fret ratio shifted back. The protein was, in effect, a genetically encoded calcium reporter, descended from the original Aequorean idea, but built entirely out of GFP variants. It was the story that had started with Aequorea's calcium-triggered blue converted to green by GFP, ended with engineered GFPs converting calcium to color again, this time inside any cell of any animal anywhere on Earth. The color wheel did not stop at green and yellow. The Aequorea scaffold by itself could not easily be pushed into the red. The chromophore chemistry of HBI, no matter how aggressively mutated, would not, on its own, support emission much beyond the yellow-green. To go red, biologists needed a different scaffold. They found one in 1999. A team of Russian researchers, led by the Lukyanov group in Moscow, cloned a red fluorescent protein from a coral of the genus Discosoma, a non-bioluminescent anthozoan. The cloned protein, named DSR-Red, glowed deep red with absorbance and emission peaks at 558 and 583 nanometers. It was immediately valuable, but also immediately problematic. Wild-type DSR-Red was a tetramer. 4 of the protein had to assemble into a complex before any of them could fluoresce. This made it useless as a fusion tag because tagging a protein of interest with ds-R-red would force 4 of the target protein to oligomerize together, which usually destroyed the function of the target. Tsien's laboratory took the ds-R-red scaffold and, through several rounds of directed evolution, broke the tetramer interface, residue by residue. The result, in 2002, was monomeric ds-R-red, MRFP. It was the first usable red fluorescent fusion tag. Its quantum yield was modest, but it worked. It folded as a single protein. It could be attached to any other protein and behave like GFP, only red. From MRFP, Tsien's group, with the trainee Nathan Shaner doing much of the engineering, generated, between 2004 and 2007, a whole orchard of red and orange variants. They named them, with deliberate playfulness, after fruits. M. cherry, deep cherry red, became the most widely used. M. strawberry, bright pink red. M. banana, yellow orange. M. orange, exactly as advertised. M. tangerine. M. grape. M. plum. Each was a slight modification of the chromophore environment. Each had a slightly different emission. Each could be paired with cyan, green, or yellow GF. By 2007, a single confocal microscope could acquire 4 or 5 distinct fluorescent channels in a single living cell, each labeling a different protein, each glowing its own color, all from the same 11-stranded beta-barrel scaffold first cloned out of a Pacific medusa. Meanwhile, the natural world had been quietly providing more starting material. GFP-like proteins, it turned out, were not unique to Aequorea. They had evolved many times across the tree of life. They were found in corals, in sea anemones, in zoanthids, in copepods, in lancelets, in certain jellyfish species other than Aequorea. The fluorescent protein family was, ecologically, much wider than the original Pacific medusa story suggested. The 11-stranded barrel was, evidently, a structural solution that the ocean had discovered repeatedly. Some of these natural variants turned out to be brighter than anything that had been engineered from Aequorea. The marine copepod Pontella mimocerami, for example, expresses a GFP-like protein with a quantum yield of 0.92, nearly twice as bright as EGFP. Other copepod GFPs, anthozoan GFPs, and lancelet GFPs added to the natural palette, providing scaffolds with different folding kinetics, different oxygen requirements, different photostabilities. Some were bright, some were dim, some matured fast, some matured slowly. The protein engineering community, by the early 2000s, was no longer working with a single jellyfish protein. It was working with a whole ocean's worth of variants, each tuned by evolution for some marine purpose nobody yet entirely understood. And what of the engineer himself, the man whose laboratory had carried out so much of this work? Roger Tsien was born in New York in 1952. His father was a Chinese-American mechanical engineer. His mother was an immigrant from Hangzhou. He grew up in Livingston, New Jersey. He suffered from severe asthma as a child, which kept him indoors and turned his interest, almost by default, towards a basement chemistry laboratory. At 16, he won first prize in the nationwide Westinghouse talent search with a project on how metals bind to thiocyanate. He went to Harvard on a National Merit Scholarship. He graduated summa cum laude in chemistry and physics in 1972, was elected to Phi Beta Kappa as a junior, and was described by his roommate. He went to Cambridge as a Marshall Scholar and earned his PhD in physiology in 1977, with a thesis on the design and use of organic chemical tools in cellular physiology. He returned to the United States, took a faculty position at UC Berkeley in 1982, and moved to UC San Diego in 1989, where he remained for the rest of his career. This was the kind of biography that produced a kind of scientist who could move easily between organic chemistry and cell biology, between protein engineering and instrumentation design, between rigorous biophysics and elegant practical applications. Tsien held more than 100 patents by the late 2000s. He co-founded several companies. He trained an entire generation of fluorescent protein engineers, including Atsushi Miyawaki, Alice Ting, Nathan Shaner, and many others who would go on to lead their own laboratories. He was, by any reasonable measure, one of the most consequential biochemists of his era. He was not, however, the only consequential biochemist in the GFP story. Without Osamu Shimomura's million jellyfish, there would be no Aequorea and no GFP at all. Without Douglas Prasher's clone, there would be no transferable gene. Without Martin Chalfie's worm, there would be no proof that the protein folded outside its host. The story is collaborative in the proper old-fashioned scientific sense, a chain of small, careful contributions, each built on the previous one, each unable to do its work without those who came before. In the early 2000s, the cell biology community began to suspect, openly, that this collaborative chain was on the way to a Nobel Prize. The protein had been transformative. The applications had multiplied. The color wheel was complete. The honor was due. The only question was who, exactly, the prize would honor, and when. From the lab benches of San Diego and New York, from the marine biology classroom at Friday Harbor, from the small white office at Woods Hole, where an aging Shimomura still came in to sort microscope slides, from the courier vans of Huntsville, Alabama, where Douglas Prasher had, by the mid-2000s, taken a job as a courtesy shuttle driver to support his family. The four men whose work had built GFP into the universal cellular tool waited, the way scientists wait, by getting on with whatever came next, while the slow Stockholm... And then, on a cold morning in early October of 2008, 3 telephones rang almost simultaneously in 3 different cities on 3 different continents. The phone call from Stockholm came on the 8th of October 2008, at the hour when most of Europe was just sitting down to breakfast. In Falmouth, Massachusetts, where Osamu Shimomura had retired some years earlier and continued, when his health allowed, to come into the Marine Biological Laboratory at Woods Hole, the time was just before 5 in the morning. Shimomura was 80 years old. He had lived through the war. He had lived through the bomb. He had lived through 6 decades of patient organic chemistry and 1 million jellyfish, and many cold mornings on the Friday Harbor dock. He had, by his own account, given up some years earlier on the idea that the work of his career would ever receive a Nobel. The phone rang. It was the Royal Swedish Academy of Sciences. They wanted to inform him that, in a few hours, in Stockholm, the Academy would announce that the 2008 Nobel Prize in Chemistry had been awarded jointly to Osamu Shimomura, Martin Chalfie, and Roger Tsien for the discovery and development of the green fluorescent protein GFP. The announcement, when it came at 11:45 Central European Time, was clear and unadorned. The official press release of the Royal Swedish Academy of Sciences read, "The Nobel Prize in Chemistry for 2008 rewards the initial discovery of GFP and a series of important developments which have led to its use as a tagging tool in bioscience." By using DNA technology, researchers can now connect GFP to other interesting but otherwise invisible proteins. This glowing marker allows them to watch the movements, positions, and interactions of the tagged proteins. 1/3 of the prize went to Shimomura, 1/3 went to Chalfie, 1/3 went to Tsien. The cash award that year was 10 million Swedish krona in total, about $1.5 million divided 3 ways. The medal, of course, was the same. In Falmouth, after the call, Shimomura did what he had always done. He went to work. He came into the laboratory at the Marine Biological Laboratory. He sat at his desk. He looked at his fish tank setup of small, dim glass jars from a long-finished experiment. He told a colleague that he was, primarily, surprised. He told a journalist later that the citation made him happy because it would remind people that the original work had been done on a real animal, in real cold water, on a real wooden dock, by people with real notebooks. The Pacific jellyfish, he said, deserved more credit than it usually got. In New York, Martin Chalfie was, by his own account, sleeping when the phone rang. He had not been told to expect it. He answered, listened, hung up, and went immediately to his computer to check whether the call was real. He logged on to the Nobel Prize website. He saw his own name there, alongside Shimomura's and Tsien's. He went, after a few moments of trying to absorb the news, to wake his wife, the cell biologist Toole Hazelrig, who is herself a long-time GFP researcher, and the person who, with him, had been one of the early users of the protein in C. elegans imaging. The two of them, by some accounts, took a few minutes to recover. In La Jolla, California, Roger Tsien received his call earlier. California is 9 hours behind Stockholm. He had, by some accounts, also been asleep. He was 65. He had been a Howard Hughes Medical Institute investigator for 20 years. He had won the Wolf Prize in Medicine in 2004. He had been, throughout the early 2000s, on most of the Nobel Watcher shortlists. He had, on this particular morning, gotten the call. All 3 men flew eventually to Stockholm. The Nobel ceremony took place on the 10th of December in the Stockholm Concert Hall. The 3 of them, dressed in formal black, accepted the medals and the diplomas from King Carl XVI Gustaf of Sweden. Shimomura, the eldest, gave his Nobel lecture in person. Chalfie gave his, Tsien gave his. The lectures between them walked the audience through the chemistry of bioluminescence, the structure of the beta barrel, the cloning of the gene, the engineering of the color variants, and the present-day uses of the protein in cell biology, neuroscience, and clinical research. In the press conference photograph that the Academy released, taken on the 7th of December at the Royal Swedish Academy of Sciences, Tsien, Chalfie and Shimomura stood together with the 2 physicists who had won the year's physics prize and with the literature laureate Le Clésio. It was a small, quiet group. They looked mostly like older scientists in slightly oversized jackets, doing what scientists do at press conferences, answering questions politely, pointing at slides, smiling small, careful smiles. The Nobel Award was not Shimomura's only honor of 2008. The same year, The government of Japan awarded him the Order of Culture, one of the highest civilian honors of the country. He was named a Person of Cultural Merit. In 2012, he and his collaborators received the Golden Goose Award, given by the United States Congress for unexpectedly transformative basic research. Recognition, late in his life, of how a project that had appeared to most observers in 1961 to be hopelessly obscure had ended up reshaping much of cellular biology. He was elected to the United States National Academy of Sciences in 2013. The government of Japan awarded him a posthumous junior third rank in 2018 after his death. He was, by then, an unusual modern figure, a Japanese organic chemist whose career had begun in the rubble of post-war Nagasaki, who had spent 50 years on the chemistry of glowing creatures, who had collected 1 million jellyfish from a single small bay in the upper San Juan Islands, and who had ended up wearing a black dinner jacket in a concert hall in Stockholm at the age of 80. There was, however, a fourth scientist in this story. We have already mentioned him more than once. We owe him, by the slow standards of this evening, a fuller portrait. Douglas Prasher, who had cloned the GFP gene in 1992 and sent the cDNA generously to Chalfie and to Tsien when his own funding had run out, was not on the Nobel roster. The Nobel Prize is, by its constitution, awarded to a maximum of 3 living individuals per category. The Academy had elected to recognize Shimomura for the original discovery, Chalfie for the proof of expression, and Tsien for the engineering. Prasher was not eligible for inclusion. He had, by then, also not been in the field for some time. He had left academic research in the late 1990s after several grant applications had failed. He had taken work outside science. By the time of the Nobel announcement, he was driving a courtesy shuttle bus for a Toyota dealership in Huntsville, Alabama, taking customers from the dealership to local hotels and back. The salary was modest. He was, by all accounts, doing it with the same generosity he had always brought to his scientific career. When the Nobel was announced, journalists tracked Prasher down. He spoke to them with grace. He told them, more than once, that he was happy for the laureates. They had taken his clone and, with it, done extraordinary work. He had no bitterness. He had no claim. He simply told the story of how he had, in 1992, finished cloning the GFP gene. How, when his funding had run out, he had thought it was important that the gene should not be lost. How he had sent the cDNA to several laboratories that he thought could carry the work forward, including Chalfie's, and how, after that, his own scientific career had wound down for unrelated reasons. Both Chalfie and Tsien, in their Nobel lectures, made a point of crediting Prasher's contribution. Chalfie invited Prasher and his wife to come to Stockholm at his own expense. Prasher attended the Nobel banquet. He stood with the laureates in some of the photographs. There was a small public consensus, particularly in the popular science press, that Prasher's situation reflected something larger and uncomfortable about how scientific funding had treated the people who do the actual cloning work in the dark before the headlines arrive. By 2010, in part because of the publicity around the Nobel, Prasher had returned to scientific work, first at UC San Diego in Tsien's laboratory and later at other institutions. The arc of his career bent slightly back into the field he had helped to build. No Nobel ceremony, however graceful, can entirely soften the edges of a story like this. 3 men received the medal. A 4th man, whose work had been essential, did not. The literature on GFP credits Prasher's 1992 paper as the foundational cloning work, and it is impossible to read the chronology of the field without seeing Prasher's name on the first page. But the medal, by Nobel rules, only goes to 3. In Stockholm, then, on the 10th of December 2008, 3 men in tailcoats stood on a stage and accepted a medal each. The orchestra played, the audience applauded. The king handed each man a small velvet box. Inside each box was a heavy gold disc, embossed with the profile of Alfred Nobel, with the year and the laureate's name engraved on the rim. Outside, in Stockholm, in Falmouth, in La Jolla, in New York, in Stockholm, in tens of thousands of cell biology laboratories around the world, the science went on. Microscopes lit up, worms glowed, mice fluoresced. Cancer cells lit up green and red and cyan and yellow under fusion protein tags. Calcium sensors flashed inside neurons. Synaptic vesicle fusions blinked in the cortex of awake mice. Embryos developed in time-lapse, glowing softly under low blue light, while researchers in dark rooms watched cells divide one by one. The lantern that biology had finally learned to hold was now, it seemed, in every laboratory. And a courtesy van rolled past in Alabama with a kind, quiet, "To understand what the medal honored, we first need to understand what the lantern let us see." This is, in some ways, the most surprising chapter of the story, because the medal is the part most people remember, and the lantern is the part that actually changed biology. The Stockholm ceremony lasted an evening. The work that the medal honored continues every day in every laboratory in the world that studies how cells live, how cells move, how cells decide what to become. The lantern, in other words, has done its real work after the headlines went quiet. Let us walk through, slowly and gently, what the green fluorescent protein has actually let biology see. We will not be exhaustive. There would be no end to it if we tried. We will pick a small handful of windows that the lantern has opened, and we will look through each in turn, the way one might walk along a long hallway in a dark museum and stop at each lit display. The first window is gene expression itself. The earliest practical application of GFP, the one Chalfie's 1994 paper had demonstrated, was as a reporter for whether and where a particular gene was being expressed. Take any gene of interest. Take its regulatory sequences, the promoter, the enhancers, the upstream and downstream regions that decide when and where the gene is transcribed. Replace the gene's coding sequence in a transgenic construct with the coding sequence for GFP. Inject this construct into the genome of a worm, a fly, a fish, a mouse. Wherever the regulatory sequences would normally turn the original gene on, they now turn GFP on instead. The cell glows. This is, in a single trick, an enormously powerful piece of biology. Until GFP, the standard way to find out where a gene was expressed was to kill the animal, fix its tissue, slice it, and stain it with something: an antibody, a labeled probe, a histochemical reagent. The work was laborious, and the animal was dead. With GFP, the same question becomes: turn on the microscope, find the green. The animal is alive. The signal is in real time. The signal can be tracked over hours or days as the animal develops, as a tumor grows, as a regenerating tissue heals. A second window is fusion proteins. A fusion protein is a single chain of polypeptide built by joining the coding sequences of two genes end to end, so that the cell translates the combined. If the two genes encode a target protein and GFP, the resulting fusion protein is the target protein with a glowing tag attached. The cell builds the target. The cell builds the GFP. The two come out as one molecule. Wherever the target protein goes, the green follows. This is the technique that has, perhaps more than any other, changed how cell biology is done. With GFP fusions, biologists can watch a transcription factor move from cytoplasm to nucleus when a hormone arrives. They can watch a kinesin motor protein hop along a microtubule. They can watch a viral capsid assemble inside a cell. They can watch a histone protein replace itself on chromatin during DNA repair. They can watch a synaptic vesicle protein cluster at the active zone of a synapse, then disperse after the vesicle releases its neurotransmitter. They can watch, in short, almost any protein in almost any cell in real time in living tissue. A third window is split GFP. This is one of those clever inversions that protein engineering specializes in. Take the GFP barrel and split it into two pieces. A large fragment containing about 10 of the 11 beta strands and a small fragment containing the missing one. By themselves, the two fragments do not fluoresce. The barrel cannot close. The chromophore cannot mature. Bring them together, however, and the barrel reassembles. The chromophore matures and the green appears. Now, fuse one fragment to one protein of interest and the other fragment to a second protein of interest. If the two proteins meet inside the cell, if they bind, if they dimerize, if they cluster on the same membrane, the GFP fragments meet, the barrel closes, and the cell glows. If they do not meet, no light. The protein in this configuration becomes a binary reporter for protein-protein interactions. Researchers use it to test which proteins bind which inside intact cells in real biological conditions. A fourth window is genetically encoded sensors. We have already mentioned the chameleon calcium sensor, designed in Tsien's laboratory by Atsushi Miyawaki in the late 1990s. The same FRET principle has been used to build sensors for many other intracellular signals. Cyclic AMP for protein kinase activity, for tension on cytoskeletal proteins, for membrane voltage in neurons. Other sensor strategies use single fluorescent proteins. The fluorine variants, for example, change their fluorescence intensity with the surrounding pH. Tagged onto the synaptic vesicle protein synaptobrevin, fluorines allow neuroscientists to watch single synaptic vesicles fuse with the plasma membrane, because the inside of a synaptic vesicle is acidic and quenches fluorine fluorescence, while the outside of the cell is neutral and lets the fluorine glow brightly. Each fusion event flashes once, brief and bright, in the dark of the dish. A single neuron, watched under the microscope, releases its vesicles in tiny green pulses. The signal of the synapse becomes visible. A fifth window is the brain. In 2007, a group of neuroscientists led by Jeff Lichtman and Joshua Sains at Harvard published a strategy they called Brainbow. The idea was characteristically Tsien school. Build a transgenic mouse in which each neuron randomly expresses, through Kree-Lock's recombination, a different combination of fluorescent proteins: cyan, green, yellow, orange, red. The result is that each neuron in the brain is labeled with a unique color, drawn from a palette of nearly 100 distinguishable shades. Slice the brain, image it. Each neuron's processes appear in their own personal hue, traceable through the dense tangle of the cortex like colored threads in a knot. The rainbow strategy and its descendants have allowed mapping of individual neurons inside intact brain tissue at a resolution that no traditional staining method could approach. A sixth window is animal medicine and cancer biology. GFP-labeled cancer cells are routinely injected into experimental animals to study how cancer spreads. The fluorescent cells can be tracked with whole-body imaging as they leave the primary tumor, enter the bloodstream, lodge in distant tissues, and form metastatic colonies. Each step of the metastatic process can be watched, quantified, and dissected. Drugs that block any step of the process can be tested against the live progression of the disease. The metastasis problem, which had been almost entirely a fixed tissue problem before the late 1990s, became, with GFP, a real-time imaging problem. Tsien himself, in the last decade of his career, took this idea further. His laboratory engineered fluorescent peptides intended to mark cancer cells in living tissue during surgery, allowing surgeons to see on the operating table the boundary. The peptides were attached to fluorophores that could be excited by handheld surgical lasers. The technique was tested in mice. The early results were sufficiently encouraging that clinical trials were initiated. The dream that a surgeon, looking through goggles in an operating room, could see the cancer glow against a dark background became, by the late 2010s, an actual experimental reality. A seventh window is unexpected. It is the world of consumer biology. In 2003, the United States company Yorktown Technologies began selling to home aquariums a transgenic zebrafish carrying the GFP gene under a muscle-specific promoter. The fish glowed faintly green under regular ambient light and brightly under blue or ultraviolet light. They were sold under the brand name Glowfish. The original purpose of the transgene had been pollution monitoring. The idea was that the fish could be deployed in waterways to detect environmental toxins by switching the protein on when stressed. The pollution monitoring application never quite became commercial. The pet store application, however, did. Glowfish became the first commercially sold genetically modified pet and remain in pet stores today in a range of color variants extending from green through yellow to red. An eighth window, and the last we will linger over tonight, is the world of art. In the year 2000, the Brazilian-American artist Eduardo Koch commissioned a French laboratory to produce a green fluorescent rabbit. The rabbit, named Alba, was an albino, with the GFP gene introduced into the germline. Under blue light, her white fur glowed faintly green. Alba, who lived for several years and died in 2002, was the centerpiece of an art project that Koch called GFP Bunny. The project provoked, perhaps unsurprisingly, an enormous and extended controversy about the ethics of bioart, the welfare of transgenic animals, and the relationship between scientific tools and aesthetic practice. The controversy, regardless of where one falls on it, helped push GFP into the public conversation. The protein had crossed, by the early 2000s, out of the laboratory and into the cultural imagination. Green fluorescent mice, of course, were already old news by then. They had been engineered as early as 1997 and were now being used in laboratories on every continent for studies of stem cells, of immune cell tracking. Green fluorescent pigs were bred at National Taiwan University in the mid-2000s. A Japanese-American team produced fluorescent cats, intended as model organisms for HIV research. In 2009, a South Korean team at Seoul National University bred the first transgenic dogs, a litter of beagles known as ruppy, who glowed faintly red under ultraviolet light. The fluorescent gene in the ruppy dogs came not from a jellyfish, but from a sea anemone. But the principle was identical, and the lineage was traceable, in the literal scientific sense, back through the color engineering tradition that had begun with Qien's S65T mutation in 1995, and before that, with Prasher's clone, and before that, with Shimomura's bucket. In the more workaday world of pharmaceutical research, GFP and its derivatives are now standard tools in tens of thousands of laboratories worldwide. They are used to validate transfection efficiency in cell culture. They are used as cell viability reporters in cryobiology, where the correlation between GFP fluorescence and the older trypan blue dye is, by some measurements, on the order of 0.97. They are used to mark transfected cells in flow cytometry. They are used to follow viral entry, single virus tracking, single molecule trajectories, neuronal action potentials. They are used in agriculture to mark crop traits during breeding programs. They are used in food safety to test for bacterial contamination of processed foods. The single small barrel from a single Pacific medusa, in other words, has become, in the 30 years since Chalfie's 1994 paper, a kind of universal flashlight for biology. Wherever cells go, GFP can follow. Wherever proteins move, GFP can track them. Whatever signals fire inside a cell, sensors built around GFP can read them. The protein has folded itself into the entire methodological vocabulary of the life sciences. To remove GFP and its derivatives from modern cell biology would be like removing the printing press from publishing. The discipline would still exist, but everything in it would slow to a crawl, and most of the questions that researchers ask today would simply not be askable. And what about the jellyfish itself in all this? This is the painful and quiet part of the story. The Aequorea victoria population around Friday Harbor by the early 1990s, blooms that had once provided Shimomura's collaborators with thousands of jellyfish in a single afternoon had thinned. By the early 2000s, the bay was largely empty of crystal jellies. Most modern GFP work, of course, no longer requires fresh jellyfish. The gene is in plasmids, the protein is engineered, the natural source is no longer needed. But Aequorea, in its original Pacific home, has nonetheless gone scarce. Whatever combination of climate change, ecological shifts, and ocean conditions caused the decline is not entirely understood. The jellyfish that gave biology its lantern is, in its native bay, harder to find than it once was.
It is sometime after midnight now, on the same cold Pacific coast where this story began, and the lantern is in every laboratory on Earth, except for the bay where it was first found. This is the part of the evening where the light goes very low. The bay at Friday Harbour is dark. The wooden field station, more than 120 years old now, still stands at the eastern edge of the harbour, with its small wooden boats tied up at the dock and its quiet collection of seawater tables and small laboratories inside. The water is cold. The kelp is breathing. The pebbles along the Wrack line are wet. The crystal jellies, however, are mostly gone. Where, in the summers between 1961 and 1988, Osamu Shimomura and his collaborators could collect tens of thousands of jellyfish in an afternoon, the modern visitor to Friday Harbour will, on most evenings, find very few. The population that gave biology one of its most important tools has not, in our time, been thriving in its own native bay. The reasons are not entirely clear. Likely a combination of changing water temperatures, shifting prey availability, jellyfish-eating predator dynamics, and complex shifts in plankton communities. The data are sparse. The jellies, however, are sparse too. This quiet ecological asymmetry, the lantern thriving worldwide, the lantern bearer thinning in its home, is the hush at the center of our story. It is, in some ways, characteristic of how science extracts what it needs from a particular organism, then forgets the organism. We could mourn this. We could read it as a parable. We could, on a different evening with different lighting, build an argument out of it. Tonight is not that evening. Tonight is the lullaby version. Tonight we simply note that the bay is darker than it once was, and that the people who carried out this work are, by now, mostly gone. Osamu Shimomura died on the 19th of October, 2018. He was 90 years old. He had been ill for some time. The cause of death was cancer. He died in Nagasaki, the city near which he had grown up, the city he had been 25 kilometers from when the bomb fell, the city in whose ruined medical college he had begun, almost reluctantly, the long, strange career that would eventually carry him to Stockholm. He was buried in his home country. The Japanese government posthumously awarded him. In his last years, he had continued to come into his small office at the Marine Biological Laboratory in Woods Hole when his health allowed. He had written a memoir in Japanese, titled in translation, "Learning from Jellyfish, The Path to the Nobel Prize." He had spoken occasionally at scientific meetings. He had given his Nobel lecture in 2008 in a quiet, careful voice, walking the audience through the chemistry of bioluminescence and the patient years on the Friday Harbour dock. He had, more than once, told interviewers that the work had simply needed doing. He had not, by his own account, been a particularly ambitious man. He had been a careful man. He had been, throughout his career, a man who believed that if a question was specific enough and worth asking, it was worth taking the long way around to answer. The long way had taken him 50 years, 1 million jellyfish, and a single small protein. By the end, he was at peace with how the long way had gone. Roger Tsien died on the 24th of August, 2016. He was 64. He died unexpectedly while bicycling on a trail in Eugene, Oregon, where he had been visiting friends. He had been, until that day, in apparent good health. The exact cause of death was not officially announced. He left behind his wife Wendy, a long list of collaborators and trainees, more than 100 patents, and an entire generation of fluorescent protein engineers who continue, in laboratories from San Diego to Tokyo to Heidelberg, to carry his approach forward. His own protégés, Atsushi Miyawaki, Alice Y. Ting, Nathan Shaner, and many others, were already, by the time of his death, leading their own laboratories. They continue to engineer fluorescent proteins, to build genetically encoded sensors, to design improved cell biology reporters. The color wheel that Tsien helped to build is, 2 decades on, even more complete than it was at the time of the Nobel. Martin Chalfie is, as of the time of this recording, still active. He continues to work at Columbia University. He continues to teach. He continues to study touch sensation in C. elegans. The worm whose touch neurons he had once labeled green in 1993 on a microscope in his laboratory in New York. He has spoken often, in lectures and in writing, about the role that scientific generosity played in the GFP story, about Prasher's willingness to share his clone, about the open exchange of ideas at small marine biology stations, about the importance of allowing scientists time to follow questions that do not have immediate practical applications. The Friday Harbor of 1961, in his telling, was a kind of slow, open library where biology happened because nobody was rushing. Douglas Prasher, who had cloned the GFP gene in 1992, returned to scientific work in the years after the Nobel announcement of 2008. He worked, briefly, in Tsien's laboratory at UC San Diego. He moved on, after Tsien's death, to other research positions. He has continued to speak, occasionally and with characteristic grace, about the strange path his career took. There is no public bitterness in his accounts. There is only the quiet observation that scientific funding can be capricious, that careers can fall through cracks that have nothing to do with the quality of the work, and that the right thing to do, when one's funding runs out, is to share what one has built, so that others can carry it forward. In the years since the Nobel, dozens of major prizes, postdoctoral fellowships, and laboratory awards have included GFP in some form or other in their citation. The protein is mentioned somewhere in almost every textbook of molecular biology and cell biology now in print. The 11-stranded beta barrel that closes around its self-assembling chromophore has become one of the most familiar protein structures in modern biochemistry. Generations of graduate students learn it as a paradigmatic example of autocatalytic chromophore formation. Generations of postdoctoral researchers learn it as the chassis from which fluorescent reporters of every conceivable design have been built. The protein is, by any reasonable measure, fully entangled with how cell biology now works. Which brings us, at last, to you. You have been listening this evening for a long, quiet stretch of time. You have walked with me from a Wrack line at Friday Harbor in the late summer of 1961 to the structure of a small chemical machine inside an 11-stranded beta barrel, to a graduate student's worm, to a phone call from Stockholm. You have heard the names. You have, perhaps by now, forgotten some of them. That is fine. The names are not the point of this kind of evening. The point of this kind of evening is that biology, very occasionally, learns how to make a small thing visible, not by inventing it, by noticing it. The light at the rim of an Aequorea bell was a real light. It was there before anybody knew it was there. It would have been there, pulsing soft green, twice in the dark of the bay, whether or not anybody had ever looked. What changed, slowly, over half a century, was that some of us learned how to look. And then, having learned how to look, we learned how to take the light apart. And then, having taken it apart, we learned how to put it into other living things, so that other living things could carry small lights around inside them too, for our quiet, patient inspection. This is, in a way, what biology has always done. We do not, as a species, build the machinery of life. The machinery was already there before us. What we do, when we are at our best, is hold a steady, gentle hand to it, watch it work, and learn how to translate what we see into a form that other people can also see. The translation, when it goes right, is not a small thing. The translation is, in fact, almost the only thing biology knows how to do. But as you drift toward sleep, you do not need to remember any of this. You can let the names go. You can let the molecules go. You can let go of the dates, the citations, the mutation codes, the prize ceremonies, the courtesy van in Alabama, the wooden dock at Friday Harbor. None of it is yours to hold tonight. The work has been done. The work has been written down. The work is in books and in journals and in laboratories and in the slow, quiet ribbons of green that still appear very occasionally around the rim of any Aequorea medusa lucky enough to be still drifting in the cold Pacific shelf. What you can hold instead is the smaller picture. A bay at low tide, a wrack line of pebbles and kelp, a small, near-transparent shape in the water, almost invisible, pulsing pale green twice, then gone. A man in rubber boots on a wooden dock in the late summer of 1961. A bucket of seawater, a patient notebook, a long, slow series of decades, a medal in a velvet box. A fourth man, generously, gracefully, accepting the way the story landed. 3 men in tailcoats accepting the medals on a stage. Snow, a slow microscope room somewhere on the other side of the world, where, even now, somebody is watching a single neuron glow faintly green in a slice of mouse cortex, just because, decades ago, somebody else noticed a jellyfish. This is the kind of story biology tells when nobody is rushing. It is gentle. It is slow. It is, in the end, mostly about patience. Patience to walk back to the dock the next morning and start again. Patience to crush another batch of medusae. Patience to chase a protein through one more column. Patience to wait for an oxygen molecule to find its way into the center of a folded barrel and complete the chromophore that, half an hour later, will start to glow. Patience, ultimately, to keep looking at one small green pulse in the dark, in the cold, twice, in the late summer of a year that nobody would otherwise remember. The bay at Friday Harbour is quiet now. The water is cold. There are very few jellyfish left in this particular bay, but they still exist in other bays, on other coasts, drifting through other cold waters as they have always drifted. Somewhere tonight in the dark, a small, near-transparent shape is pulsing soft green at its rim, twice, and going still. Nobody else sees it. The pulse happens whether or not it is observed, the way the tide happens whether or not it is measured, the way light happens whether or not anyone is looking. Let that be the picture you carry into sleep. A small, soft green light in a cold, dark bay, pulsing twice, then gone. A whole science wrapped around it, gently. A whole laboratory life wrapped around the science. A whole human history wrapped around the laboratory life. And at the very center of all of it, the small medusa itself, doing what medusae have always done, which is to drift gently at the level surface of the sea, almost entirely water, almost entirely transparent, glowing once or twice when the night is right, asking nothing of us, owing nothing to us, simply being itself. The dock is empty, the water is dark, the kelp is wet. The lantern is on somewhere in 1,000 laboratories, even tonight. The jellyfish is doing what jellyfish do. The story has been told. The light is steady. You are warm where you are. You are safe where you are. You can let the names go now. You can let the dates go. You can let the bay go. Sleep well, child of the cold Pacific coast. Good night.